Good morning, Bio. Come on in and have a seat. I'm delighted to be back. I understand you guys have been just killing it in Mendelian genetics and understand how inheritance works and how variation arises and gets transmitted from parents to offspring within a family. That's great because that will all be really important stuff as we now transition to the study of population genetics. So inheritance not just within a single family, you cross this male and this female, but within a whole collection of families. The good news is that Mendelian principles still continue to apply even when we scale things up massively to the scale of a whole population. So let's see how that works. I know that probably many of you have been prioritizing the studying for the midterm, and that's fine, but raise your hand if you were able to look at Simutext and do the assigned. Okay, well, maybe after the midterm, you guys want to click through that because there's all sorts of really good practice questions and activities on the Mendelian Pigs module. But let me give you a quick overview right now. The whole Mendelian Pigs module opens with this kind of hook, I guess, or a scare story. It's this story. This is an actual story that was in the news, in the BBC Science News a while ago. And it was headlined like a tragic fate for blondes. And people were getting all bent out of shape because there was this fear that humans with naturally blonde hair are going to decrease in frequency and eventually completely disappear. The alleles associated with blonde hair coloration in humans were going to disappear by the year 2202, apparently. And people with blonde hair are an endangered species. So we'll interrogate this, see if it's really something to be concerned about, or is this fake news? The story goes on to say, if you're reading around in Simutext, that the issue with blonde hair is that blonde hair is caused by a recessive allele. So this whole news story, people getting all agitated, preassumes that the fate of an allele is linked to its dominance status. So you know about dominant alleles and recessive alleles. This story presupposes that recessive alleles, just by virtue of being recessive, are going to decrease in frequency. Conversely, that dominant alleles, like alleles associated with dark hair color in humans, are going to increase in frequency. And how likely is this? Well, it's true that a lot of very rare alleles, I'm thinking especially of rare alleles associated with genetic disorders like the cystic fibrosis allele or the Tay-Sachs allele. A lot of these very rare alleles are also recessive, and we'll talk more about why this is the case. So maybe, yeah, recessive alleles are just rare and getting rarer. On the other hand, you can probably all think of some recessive alleles that are associated with traits that are pretty common. Can anyone think of one? Yeah. 5 fingers, is that recessive? Yeah, I guess it is recessive. Yeah, that's right. There's a dominant allele associated with having 6 fingers. Okay, yeah, so there's a recessive allele that's actually very common. Raise your hand if you are blood group O, if you know your blood type. Okay, yeah, a lot of people here, that is another example of a recessive allele that's very common in humans. So is there truth to this idea that the dominant status of an allele will affect its frequency in a population? So we can, yeah, clicker question, okay. This is just one of the practice questions from Simutext. There's so many practice questions there, but get yourself set up to click if you can. And imagine that we have a dominant allele that's pretty common and a recessive allele that's pretty rare. And there's no selection, they're both equally fit. how are the allele frequencies going to respond to that? So think about that for a minute. You can talk to your neighbor if you're not sure. Okay, give it a look. All right, so most people, the most popular answer here was D, that if there's no selection, then allele frequencies probably aren't going to change. They're not going to change much. Second most common answer was we're going to see convergence onto these sort of classic Mendelian ratios, like a 3-to-1 ratio, 0.75 for the dominant allele and 0.25 for the recessive allele, because that's how Mendelian inheritance works. I'm not actually going to tell you the answer just yet. We're going to explore it using the SIN pigs. So let's see. All right. So here, we're going to work instead of, I don't know why, but this program doesn't work with people of different hair colors running around in a city. It works with these imaginary simulated pigs running around in a field. But it's the exact same gene that is involved in human hair color is also involved in pig coat color and the color of your dog's fur and the color of bird feathers. You don't have to remember the name of the gene. But for now, the important thing to know is that there are several different alleles at this particular locus. And one of them is called the B allele, B for black, and it is dominant to the R allele, R for red. Okay, so if you have 2 copies of the R allele, your homozygous R, you're going to be red, a red pig. If you have 2 copies of the B allele, you're a black pig. If you have 1 copy of each, you're also a black pig. So B is fully dominant to R. The heterozygotes have exactly the same phenotype as the homozygous dominant. You're probably very familiar with this situation coming fresh out of Mendelian genetics. So let's imagine that we have slightly more copies of the B allele than the R allele. We're going to, we can track the frequency of, say, the B allele along this axis. All I'm going to do now is let these pigs run around the field, and every time a pig bumps into another pig, it reproduces with it, and they produce offspring. And the genotype and the phenotype of the offspring depends on the genotype of the parents. And there's a lot of pigs there now. Are the allele frequencies changing much? Okay, let's stop. Okay, so here we're tracking, what is it, the frequency of the B allele associated with black coat coloration. So we see that it starts at a frequency of about 0.6, and it bounces around a little bit for reasons that we'll get into maybe in the next lecture, but it's really not changing appreciably. And likewise, the frequency of the R allele, the recessive allele in this system, starts at about 0.4 and doesn't really change over time. And so what this shows us then is that, I'm just going to pull this up here. If there's no selection, If there's no selection, that is, if all the genotypes have the same fitness, in other words, if there's no selection happening, then it doesn't matter whether an allele is dominant or recessive, or that's not going to change the frequency of that allele. So that BBC story, so the red pigs aren't going extinct, the R allele is not disappearing from the population. By extension, people with blonde hair are not going to decrease in frequency simply because blonde hair is associated with a recessive allele. Fake news, BBC, fake news. We can ask, though, if the genotypes did not have the same fitness, can the dominant status of an allele be important in that case? In other words, do alleles that are dominant versus recessive, might they respond in different ways to selection? You might say, how am I going to impose selection on this population? Well, the program can do it. So let's imagine now that we've got pigs. Oh, so many pigs in this field. I'm going to reset them. We've got black pigs, and we've got red pigs. And we can start with equal numbers of each, equal allele frequencies, or we could start with, do you guys want to start with one color being more common than the other, or 50-50? I don't care. 50-50? Okay, all right. So we've got black pigs, we got red pigs, but now we're going to introduce selection. Okay, so first of all, let's imagine that, okay, you see these wolves here? These gray things are supposed to be wolves. Imagine that there are wolves in the field that only like to eat black pigs. Okay, so now we have introduced selection against the B allele, or the black genotype and phenotype. So we can introduce a death rate from wolves, and we could, you guys want to see strong selection or weak selection? Strong? Okay, all right. So we'll say like 25% of the black pigs get eaten by the wolves. Okay, so that's how I do that. I introduce a death rate from wolves of 0.25 is the fraction of black pigs that get eaten. The red pigs are going to be fine. So their death rate from wolves is going to remain 0. And now, we're just going to let this go and track the, okay, let's track the frequency of the B allele, which is now like a harmful or a deleterious allele that's being selected against. And let's see it go. First, do you guys think that the B allele, this harmful allele, is going to go down in frequency or up in frequency or stay the same? Obviously, it's going to go down. Yeah, okay. Okay, so the frequency of the B allele is going down. You see there's more and more red pigs along the bottom. This is pretty strong selection we've introduced. And eventually, there's still some black pigs in the mix, but the wolves keep on finding them and eating them. And eventually, no, not yet. Okay. Frequency of B is not quite at 0. Look at that. It's still, every now and then, a black pig will pop. Oh, no, okay. Now, okay, the black pigs are all gone. Okay, the B allele, this harmful dominant allele, has fully disappeared from the population. That is what happens when we have selection against a harmful dominant allele. And does that make sense to everybody? Okay, good. However, we could, just for the sake of argument, say, what if the wolves weren't preferentially eating the black pigs? What if they were eating the red pigs? So what if the B allele is actually beneficial? It's good to be a black pig, and it's bad to be a red pig. So if the recessive allele is harmful, or deleterious, that's another word for harmful, or the red allele, the recessive allele, is being selected against. So what we'd do to model that is say, okay, now the black pigs are fine, and the red pigs have like super strong selection. against them. Okay, and so we'll put in a bunch of red pigs and a bunch of black pigs like that, and yeah, and we'll go, sorry, I'll track the R allele now. So now the R, recessive R allele associated with the red coat coloration, it's now the harmful allele. And as we would expect, like we saw with the selection against the B allele, it is decreasing. The harmful allele is decreasing in frequency. That's what selection does. Still a few red pigs, not very many. Frequency of the R allele is getting pretty low. Do you think the frequency of the R allele is going to go all the way to 0? No, why not? Yeah. Yeah, pretty much. Because the harmful allele in this case is recessive, some individuals are still carrying a copy of the red allele, but they're heterozygous. And so it's still a black pig, but it's in a heterozygote, and so that individual is fine. So these harmful recessive alleles can still survive and persist because they are like sheltered in the phenotype of heterozygotes. Whereas if we have a harmful dominant allele, selection is going to be able to see every single copy of that dominant allele, whether it's in a homozygote or a heterozygote. So in this case, this is a subtle but an important difference between selection against a harmful dominant allele, which fully removes the bad dominant allele from the population. Selection against a harmful recessive allele makes it decrease in frequency, but almost certainly not all the way to 0. And that's because the recessive allele can still persist in heterozygotes. So because these pigs are all diploid. And this, of course, is the reason that the vast majority of genetic disorders are associated with recessive alleles. It's not that recessive alleles are just innately trash or bad alleles or reduce fitness. It is because if there was anytime a genetic variant pops up that reduces fitness and is like a dominant allele, selection is going to just fully remove it from the population. Selection cannot remove every harmful recessive allele from the population. And so that's why genetic disorders are typically associated with recessive alleles. Does that make sense so far? Okay, amazing. Because it's going to get a little bit more complex now. No, not more complex, just a little more fun. Okay. So up until now, we've just been dealing with a relatively simple system. There's one allele that's dominant, and there's one allele that's recessive, and the heterozygotes have the same phenotype as the homozygous dominants. Now, you probably know that not all alleles and not all genes, not all loci work that way. Sometimes we can have incomplete dominance or co-dominance. That is, sometimes we can have systems where the heterozygotes have a different phenotype from either type of homozygote. So if there's 2 alleles at a locus, we might have like 3 possible different phenotypes. Like, one type of homozygote is red, and one type of homozygote is pink, and one type of homozygote is white, and the heterozygote is pink, or is like red and white stripes or something. Anytime that heterozygotes have a distinct phenotype from either type of homozygote, it's possible that heterozygotes could also have their own distinct fitness. And a great example of that is what we call heterozygote advantage. So imagine if the wolves were not preferentially eating, this type of homozygote or that type of homozygote, but what if the heterozygotes had higher fitness than either type of homozygote? We can model that in the Simutex system by introducing another allele. So you already know the R allele, and if you have 2 copies of R, you're a red pig. Another allele in the system at this locus is called the S allele. If you have 2 copies of the S allele, homozygous S, you are red with a lot of spots. If you have 1 copy of S and 1 copy of R, that is, if you're a heterozygote, then you have kind of an intermediate phenotype. You're red with a few spots. And if you have no copies of S, just 2 copies of R, then you just don't have any spots. Okay, so heterozygotes have a different phenotype from either of the homozygotes. And we can imagine a situation where this heterozygous phenotype is going to have the highest fitness. So if the wolves like to eat say we have lots of spotted pigs and a few red pigs. And now let's say that the wolves like to eat the red pigs, but they also like to eat the heavily spotted pigs. But they're going to leave these guys, these heterozygotes, red with few spots, they're going to leave them alone. Why? Why do the wolves have this preference? I don't know, but we're just making this up for the sake of argument, yeah? So now we can trace, let's say, we'll trace the frequency of the S allele. We're going to start with a lot of copies of S, lots and lots and lots of copies of S, and we're going to let this go and take a look. So we're starting off with the big S allele, the S allele being very common in the population, and it's starting to decrease. Okay, yeah, and we can see the frequency of the R allele in that bar and the S allele. And you can see that R started off as rare, but it's starting to increase in frequency. S started off as common, it's decreasing in frequency, until eventually they both hit about 0.5. And at that point, this allele frequency is going to basically level out. I'll let it run for a few more seconds just to convince you of that, or you're welcome to play around with the Simutext on your own device at some point after the class. So there we go. Okay, let's stop this. Good job, pigs. Okay, so this is what happens under heterozygote advantage. So we started off with 2 alleles in the population. Heterozygote advantage maintains both of those 2 alleles. Heterozygote advantage maintains genetic variation. Neither of those alleles is going to go extinct. And I know, I think we started this with lots of copies of S and not very many copies of R, but it actually wouldn't matter. We could have just flipped the starting frequencies and had like a bunch of red pigs and only a few heavily spotted pigs, lots of copies of R and only a few copies of S. And again, the common allele would have decreased in frequency to 0.5. The rare allele would have increased in frequency to 0.5. And both of them, once they hit 0.5, that's where they're happy. That's their equilibrium frequency. So starting allele frequencies don't matter. We're going to wind up at exactly the same place, no matter which allele starts as common and which allele starts as rare, under heterozygote advantage. I want to just hop real fast to a clicker question to test your understanding of this so far. Okay, so heterozygote advantage, here's an allele frequency, and it starts off common and then it decreases until it hits 0.5. Is this selection? Did we just demonstrate selection happening? What do you think? So the best way to think about selection is not all genotypes having the same fitness. So if the different genotypes vary in their fitness, we would say that selection is happening. And so the correct answer to this is A. Selection is happening, and is happening this whole time. Even after like 1,000 generations of pigs, after the allele frequencies have kind of stabilized, the heterozygotes still have that advantage. That is still the fittest of the genotypes. So selection is still happening. Selection is happening this whole time. Selection is just genotypes having different fitnesses from each other. What if I didn't ask you about selection? What if I asked you about evolution? Is the population evolving? And remember the relationship between selection and evolution. So is the population just not evolving at all? Is the population evolving for a while, like while the allele frequencies are changing? Or is the population evolving the whole time because selection is happening the whole time? Think about it. Going to show me the answer. Maybe it is. Okay. Is the population evolving? The correct answer here is B. Evolution was happening for a while. Evolution is a change in allele frequencies. After the allele frequencies hit 50-50, they're basically not changing at all. So evolution was happening for a while. Evolution is not happening towards the end of the simulation. And collectively, when we think about those two things, yeah, question. Would that mean that the horseshoe crab is not evolving? It appears in the horseshoe crab example that it's not, at least its morphology is not changing, yeah. Whether there are various alleles that are, other alleles that are changing. Yeah, so long periods of no change, yeah, mean no evolution. Yeah. Okay, I just want to point out that collectively, the fact that towards the end of the simulation, you still had selection, but it no longer caused evolution. It means that yes, it's possible to have selection that does not result in evolution. Yes, question. Is it bad for the population if they're not changing? Not necessarily, no. The population is more fit to, it's better suited to its environment than it was at the beginning. So the environment, is remaining very stable throughout this whole simulation, and so we might not expect the population to keep on changing once it's sort of reached the best state. Yeah.
Is there a case where there's a population that's not changing because environmental factors say that, like, hey, you're well suited for this?
Yep.
Put it naturally much that there's nothing really, like, pushing that change and it's not really.
Yeah, if there would be natural selection, assuming that there's variation in the population, and there are all sorts of variants, but it's like the same phenotypes keep on getting favored. That would still be natural selection, but it wouldn't necessarily cause evolution. Just like in the pigs. Okay, you guys have so many questions today. I love it. All right, but let's talk about another. Okay, so I've told you about heterozygote advantage. And in heterozygote advantage, which is like a really important way, that it's a big reason why there is so much genetic variation maintained in natural populations. Because often it's not that there's like one good allele and one bad allele. No, it's the combination of having one of each is maybe the best. Under heterozygote advantage, genetic variation is maintained, and it doesn't matter the starting frequencies. You could start with this allele being more common, or you could start with that allele being more common. Also in heterozygote advantage, it doesn't really make sense to talk about the dominant allele and the recessive allele, because if the heterozygotes have their own unique phenotype. But the converse, or the opposite of heterozygote advantage, is heterozygote disadvantage, or homozygote advantage. Imagine the situation where we've got, again, the RR and the RS and the SS pigs, but now we're going to say that actually it's the heterozygous pigs, the red with few spots pigs, get preferentially eaten. And both the homozygotes are fine. So in this case, do we want to start with like more red pigs or more spotty pigs? Red? Okay. Red, okay. I'm going to start with lots of red pigs here. Okay, so here we're going to model heterozygote disadvantage, which is a synonym for homozygote advantage. Okay, so here we've got this R allele. It starts off common, and oh, look at that, it's becoming even more common. It's increasing in frequency. The other allele, the S allele, is decreasing in frequency. And eventually, we can let this play out. Do we think S is going to completely disappear? Still there. Still there. Oh, okay, yeah. Okay, and at this point, the R allele has completely gone to fixation. R has reached a frequency of 1, S has reached a frequency of 0. Now, what if we had started with lots of SS pigs and just a few R pigs? The what? The wolves would be full. The wolves are living their best lives. If you start with the SS genotype, or a high frequency of S and a low frequency of R, and you're welcome to run it yourselves on your own devices if you don't believe me. You should believe me, because I've played with this game many times. But it would be the S allele that rises to fixation, and the R allele that goes extinct. In other words, unlike heterozygote advantage, where the starting frequencies don't matter, both the alleles are going to wind up in the same place. The common allele is going to decrease in frequency until it's no longer common. The rare allele is going to increase in frequency until it's no longer rare. In heterozygote disadvantage, the starting frequencies totally matter. They are critically important. And whichever allele starts off with the higher starting frequency, that is the allele that is going to rise to fixation. And whichever allele starts off rare is going to decrease in frequency and eventually disappear. And it's not because that rare allele is inherently bad or inherently some trash allele. No, it's the fact that it is rare. Sometimes it is just selection against the rarer allele. I'll get more into that in the next lecture, because I know it's sort of it's sort of less intuitively easy to grasp than how heterozygote advantage works. Heterozygote advantage, heterozygote disadvantage, they have opposite effects on genetic variation. Heterozygote advantage maintains both the alleles. Heterozygote disadvantage removes one of the alleles. But they also differ in how important the starting allele frequencies are. In heterozygote advantage, it doesn't matter. In heterozygote disadvantage, it does matter. Yes? Yeah, okay. Yeah, so I love that question. What if the wolves then started paying attention and oh, there's a lot of red pigs here, we should switch to eating the red pigs. Then that would give rise to like what's known as an arms race. Like we see arms races between predator and prey. We see arms races between hosts and disease organisms. That is too complex for this little simulation to get into. But yeah, that's a great question. This whole thing assumes that the selective environment just stays exactly the same, which is not really realistic, but yes. Oh yeah, okay, so heterozygote disadvantage, and we started off with 0.5 and 0.5. That is what we call an unstable equilibrium. In theory, if the population is big enough, we could, that could maintain itself for a very long time because we don't have a common allele and a rare allele. However, you'll notice that the allele frequencies bounce around a little bit, and sometimes even just one of those little bounces just knocking the frequency of, it's unpredictable which allele is going to increase in frequency and which is going to decrease, but one is going to just bounce down to say 0.49 instead of 0.5. And then once that happens and it becomes more and more rare, then it's sort of a self-perpetuating thing. In this case, it's the S allele that seems like it's becoming more rare. In theory, if there were a million, billion, trillion pigs in this, it might bop around for a long time at 0.5, but it's a very unstable situation. Great question. Okay. Oh, that's it. That's it for the pigs. So say goodbye to them. It's too bad. Okay, and now we can start thinking more broadly. We can generalize now beyond pigs and alleles associated with coat color and just think more broadly about the frequencies of different alleles and the frequencies of different phenotypes in populations. How, it may seem so different from what you know about Mendelian inheritance, where you might have like a 1 to 2 to 1 ratio of phenotypes or a 1 to 3 to 3 to 1 ratio of genotypes, because in populations, you might have, like allele frequencies of 0.7 and 0.3 or something. You might not see these sort of classic, what we think of as Mendelian ratios of phenotypes and genotypes, but all the rules of Mendelian inheritance still apply. As long as we know the distribution of parental genotypes in like a dihybrid cross, say, we can predict the distribution of offspring genotypes. You already know how to do that from high school and from the last few cycles. In population genetics, if we know the distribution of genotypes in the population, we can often predict the distribution of genotypes in the next generation. Okay, so this is a Punnett square. This is hopefully review to you. This, if we had like one parent A mating with parent B, and both of them are heterozygotes, they have one copy of allele W and one copy of allele R, we can use, you may remember the product rule. Why do we think that it's a quarter of the offspring are expected to have this RR genotype? Because we're going to multiply 0.5, the fraction of this parent's gametes that bear that R, by 0.5, the fraction of this parent's gametes that carry the R. Both of those things have to happen, so we multiply 0.5 by 0.5. We say there's a 25% chance that the next baby is going to have an RR genotype if these are the parents. This is familiar to you. This is maybe less familiar, but this is how we can scale up a single Punnett square and apply it to a whole population. Okay, so just as if we have a parental generation and the frequencies of the W and the R allele are, say, 0.6 and 0.4, respectively, then we can use those allele frequencies to predict, say, how many offspring should have this homozygous W genotype. The likelihood is 0.6 times 0.6, or 36% of the offspring we might expect to have this genotype. The only difference between this type of analysis and Mendelian analysis within a single family is that the allele frequencies aren't necessarily 0.5 and 0.5. They could be 0.9 and 0.1, or 0.2 and 0.8 or something. Otherwise, it's exactly the same logic applying the product rule. And this idea was sort of formalized by, actually not by biology professors, but by two mathematicians who independently stumbled upon this insight that we can use we can use Mendelian rules to predict genotype frequencies in a population, at least under certain conditions. And these are very, very specific, very, very restrictive conditions, but let me go over them now. If a population is very, very large, if the population is mating randomly, so if we're talking about pigs just mate with the first individual that they bump into. They're not, we don't have red pigs only mating with other red pigs or black with black. It's just like a bunch of gametes sloshing around in a bucket. Mating is random with respect to genotype. If, furthermore, the mutation rate is so low that we can essentially ignore it, say it's very close to 0. If the population is closed to new individuals coming in or old individuals just deciding to leave, so there's no gene flow, no immigration or emigration, and if there's no selection, if and only if all of those things are true, then a couple really important things emerge. First of all, evolution is not going to happen if all of those conditions are met. The allele frequencies are not going to change. The other thing that happens, which turns out to be very useful in identifying whether or not these conditions are met, if all of these conditions are met, then the genotype frequencies can be predicted from the allele frequencies. And specifically, if we have 2 alleles at a locus, we can have more than 2 alleles at a locus, but the formula gets a little bit more complex. We have 2 alleles at a locus, and we call the frequencies of the 2 alleles P and Q, respectively. Then the expected genotype frequencies of one type of homozygote would be P squared, using the product rule, the fraction of alleles that carry P, but you need to inherit that from both of your parents, so that's why it's P times P. The proportion of individuals that we would expect to be heterozygotes would be P times Q times 2, or 2PQ. And this is because there's 2 ways of getting this A1, A2, this heterozygous genotype. You could inherit A1 from dad and A2 from mom, or you could inherit A1 from mom and A2 from dad. There's 2 ways of being a heterozygote, both the same genotype. And frequency of this other type of homozygote is equal to Q squared. These are the classic Hardy-Weinberg genotype frequencies. This set of conditions up here is very restrictive, and it also describes a situation which is really boring. There's nothing good or interesting or cool going on here in this population. Selection is not happening, and they're not exercising non-random mate choice or anything. There's not gene flow stirring things up. So why do we care about the Hardy-Weinberg equilibrium if it's boring? Who cares about a population that's not evolving if we're interested in evolution? We need to know what a population would look like if it's not evolving. And this only by knowing what to expect if no evolution is happening can we recognize a population that might be evolving. So it's like a null model. It gives us something to compare observed genotype frequencies to. And so if the genotype frequencies can be predicted from the allele frequencies in the population, if they conform to that p squared and 2pq and q squared set of terms, then the population is in Hardy-Weinberg equilibrium at that locus, and the population is not evolving at that locus. Conversely, if the genotype frequencies we see are different from the genotype frequencies that we would expect, they don't match up with p squared, and the heterozygotes do not have a frequency of 2pq, and the other homozygote does not have a frequency of q squared, then that tells us the population is not in Hardy-Weinberg equilibrium. So one or more of the assumptions of Hardy equilibrium, whether it's about mutation or migration or selection or mating, one of these assumptions, maybe more of these assumptions, are being violated. It's possible the population then is evolving. The textbook goes through in great detail how you can calculate, how you can test whether a population is in Hardy-Weinberg equilibrium or not. So if you haven't done the pre-reading for today's lecture, please do it as soon as you get a chance, maybe after the midterm. And in particular, I would encourage you to read over table 17.1 and figure 17.8. But it goes through how you can calculate genotype frequencies and allele frequencies at this particular flower color locus in snapdragon plants. But basically, you would always be given a number of, there's this many individuals with this genotype, and there's this many individuals with this genotype, and this many with that genotype. It's then just a matter of counting up how many copies of this allele, of the CR allele are there, how many copies of the CW allele there are. And the textbook goes through it all, but if people have questions with it, you're always welcome to come to my office hours and I can walk you through it as well. So the first step in solving a Hardy-Weinberg type question is to figure out what are the allele frequencies. I will give you the starting genotype or phenotype frequencies. You can count alleles basically and figure out, okay, P is equal to this and Q is equal to that. And then you just plug those values of P and Q into those Hardy-Weinberg proportions. So if it's in Hardy-Weinberg, we would expect there to be P squared of this type of homozygote and 2pq of the heterozygote and so on. And then the final step then is you've got the observed set of genotype frequencies that you would be given at the start of the question. You've got the expected set of genotype frequencies that you've just worked out. And then you just ask, are they the same? And if they are, then the population is in Hardy-Weinberg equilibrium, and nothing interesting is happening. But if they're different, in this case, we observe more heterozygous individuals with this pink phenotype than we would expect if the population were in Hardy-Weinberg. And that tells you that something interesting is going on. It doesn't necessarily tell you which of the assumptions is violated. You might need more information to get into that, but we'll talk more about that in the next lecture, how you can sort of infer which type of violations are more likely or less likely. So to summarize then, if all of these conditions are true for a population, and again, this is a very restrictive set of conditions, if that's true, we say the population is in Hardy-Weinberg equilibrium. I think your textbook calls it genetic equilibrium, but it's the same thing. And if the population is in Hardy-Weinberg equilibrium, then evolution doesn't happen, allele frequencies don't change, and the genotype frequencies can be predicted from the allele frequencies. So just an overview of what we've covered today then. Does the dominant, are blondes going extinct? No. The dominant status of an allele in the absence of selection doesn't affect its frequency. But if selection is happening, then yes, the dominant status can become important. Familiarize yourself with the different types of selection and how each of them affect genetic variation at a single locus. Some types of selection remove genetic variation, some of them maintain it. And why do we care about the Hardy-Weinberg principle, even though the absence of evolution is boring? Because you need to understand the Hardy-Weinberg principle to recognize the population that could be evolving. If it's in Hardy-Weinberg evolution, evolution is not happening. If it's out of equilibrium, maybe evolution is happening, and we'll talk more about that next lecture. Good luck to everyone on the weekend, and yeah, I'll see you on Tuesday. Thanks, gang.